In 1961, Leonard Hayflick made a discovery that challenged a basic assumption in biology. When he grew normal human cells in the lab, they didn't divide forever. Instead, they stopped after around 50 to 70 divisions. This became known as the Hayflick limit, showing that cells carry an internal brake on proliferation. We now understand this brake as part of a process called cellular senescence. Senescence is the stable and terminal state of growth arrest in which cells are unable to proliferate, despite optimal growth conditions and mitogenic stimuli. Senescence can be triggered by several forms of persistent cellular stress. The main ones are telomere shortening, DNA double-strand breaks, oncogene-induced stress, and mitochondrial oxidative damage. Although these triggers differ, they all converge on the same outcome. Continued division would be dangerous. One of the most important triggers is telomere shortening. During DNA replication, the lagging strand is copied in short Okazaki fragments, and because the final RNA primer cannot be replaced, a small portion of DNA is lost each time a cell divides, around 50 to 100 base pairs. To protect essential genes, chromosomes end in telomeres, repetitive non-coding DNA that acts as a sacrificial buffer. Over time, telomeres become critically short. At this point, the cell enters senescence to prevent genomic instability. While an enzyme called Telomerase can rebuild telomeres, most somatic cells suppress it because extending cellular lifespan greatly increases cancer risk. Another trigger is DNA double-strand breaks, where both strands of the DNA helix are severed at the same location. These are among the most dangerous forms of damage. When DNA deteriorates beyond reliable repair, senescence acts as a failsafe to permanently halt division. Senescence can also be induced by oncogenes. Overactive growth signals push cells to divide too aggressively, causing replication stress and DNA damage. Rather than becoming cancerous, cells activate tumor suppressor pathways and shut themselves down through senescence. Mitochondrial dysfunction provides another route. As mitochondria age, electrons leak from the electron transport chain, forming reactive oxygen species. These molecules damage DNA and telomeres, triggering persistent stress responses and senescence, even without excessive cell division. These trigger the DNA damage response pathway. If damage is minimal, DNA can be repaired and cell goes back to normal. However, irreparable damage causes senescence. Both paths begin with the activation of p53 and induction of the cell cycle inhibitor p21 to pause the division cycle. In senescent cells, there's the accumulation of p16, which locks the cell in a long-term senescent state, with the tumor suppressant ARF stabilizing p53. At first glance, senescence sounds entirely harmful, but it plays essential roles. It suppresses cancer, helps shape tissues during development, and limits the spread of damaged cells. Senescent cells also release signaling molecules known as the senescence-associated secretory phenotype, or SASP. In the short term, SASP factors can promote wound healing and attract immune cells to clear senescent cells once they've served their purpose. Problems arise when senescent cells accumulate and are no longer efficiently removed. Chronic exposure to SASP drives inflammation, creates tissue environments that can support tumor growth, and disrupts stem cell niches. As stem cells become exhausted, tissues lose the ability to regenerate and aging accelerates. Crucially, senescence itself is not the problem. It is the accumulation of senescent cells and the failure of clearance that causes damage. Because of this, researchers have proposed therapies that target senescence. Senolytics aim to selectively kill senescent cells, reducing inflammatory burden and relieving pressure on the immune system and stem cells. Senomorphics, by contrast, suppress harmful SASP signaling without removing the cells, making them a more conservative approach. In conclusion, aging is not a mistake. Natural selection prioritizes the passing on of genes, not the individual's health. As a result, senescence genes are strongly favored as it is heavily favorable in an individual's early life, thus promoting reproduction. Selection frankly doesn't care about how the individual lives. The genes have been passed on already. This is why, to extend our longevity, we've got to look for answers ourselves instead of waiting for nature to step in. Thank you for watching. Make sure to like and subscribe if you enjoyed the video.